Have you ever considered something as seemingly simple as fat?
Yeah, it sounds simple, right? But it's actually like incredibly complex.
Exactly. We often just lump it all together. But what if I told you that fat, well, more accurately, biological lipids are so incredibly diverse and vital?
Oh, absolutely vital. And we're still, uncovering their profound secrets
Mm-hmm. From building the very walls of our cells.
to powering our internal engines, sending crucial messages. It's amazing stuff.
Welcome to the Deep Dive, where our mission is to unearth the most important insights from curated sources. Today, we're taking a deep dive into the fascinating world of biological lipids, pulling key nuggets directly from Chapter 10 of Lehninger Principles of Biochemistry.
Right. Think of this as your shortcut to understanding their molecular mechanisms, their biochemical pathways, and really why they're so fundamental to life.
Our goal is to reveal how these amazing molecules, despite their common feature of insolubility in water, serve just an astonishing array of functions.
Yeah, and we'll try to break down some of the maybe more complex structures and reactions into understandable insights. So you walk away, feeling pretty well informed about this essential class of biomolecules.
Okay, let's jump in. If you had to pick one defining characteristic, just one thing that ties all lipids together, what would it be?
Their universal insolubility in water, that hydrophobicity, it's the foundation for pretty much everything they do.
And what a range of things. Most of us think fats and oils, energy storage. But beyond that, what other critical functions are lipids performing?
Well, they're the major structural bits of every cell membrane forming those crucial boundaries. But even in tiny amounts, they're essential as like enzyme helpers,
Uh-huh.
electron carriers in energy production, light-absorbing pigments, and really powerful chemical messengers, things driving inflammation, cell growth. It's incredibly versatile.
That's way more than just energy. Okay, so our source material emphasizes how lipids are incredible fuel, building blocks, and active signals. Let's start with the fuel part, fatty acids being so energy-rich.
Precisely. Fatty acids are basically these dense, water-insoluble hydrocarbons just packed with chemical energy. When our cells break them down, they release more than double the energy per gram compared to carbs or proteins.
Wow, double. And their stored form, these triacylglycerols, that maximizes the efficiency.
Exactly. Because triacylglycerols are so hydrophobic, so water-fearing, they don't need to carry extra water weight with them.
Unlike carbohydrates.
Right. Carbs are heavily hydrated. So storing energy as fat saves a huge amount of weight. It's a massive advantage, especially if you need to move around.
Okay, let's dig into these fatty acids a bit more. What's the basic structure, and why does that make some fats solid, like butter, and others liquid, like, say, olive oil?
Okay, so fatty acids are essentially long carbon chains with an acid group at one end. They can be saturated, meaning no double bonds in the chain,
Right.
or unsaturated with one or more double bonds. The big difference is how they pack together. Saturated fatty acids are straight. They can stack very neatly, almost like bricks.
Right, nice and orderly.
Yeah, and that strong packing makes them solid, sort of waxy at room temperature. Think butter or animal fat.
But the unsaturated ones have a kink.
That's the key. The cis double bonds in most natural unsaturated fatty acids introduce a definite bend, a kink in the chain.
So it messes up the stacking.
Totally. It's like trying to stack, I don't know, crooked logs instead of straight bricks. They just don't fit together neatly.
Okay.
That kink prevents tight packing, weakens the forces between the molecules. So less energy is needed to melt them, which is why they're typically oily liquids at room temperature, like olive oil.
That makes perfect sense. Now, we hear a lot about omega fatty acids in nutrition. Why is that omega system so important for our health?
Well, the omega system is actually pretty helpful because it tells us where the first double bond is, counting from the tail end, the methyl end of the fatty acid chain.
Right, so omega-3 means the first double bond is 3 carbons in.
Exactly. And humans, we can't actually make certain crucial omega-3s like alpha-linolenic acid or ALA. We have to get it from our diet.
So it's essential.
It is. And we use it to make other vital omega-3s like EPA and DHA. The issue is that in many Western diets, the balance is way off.
How so?
We tend to get far too many omega-6s compared to omega-3s. Sometimes the ratio is like 30-to-1, when ideally it should be closer to maybe 1-to-1 or 4-to-1.
Wow, that's a big difference. And that imbalance matters.
Yeah, it's strongly linked to an increased risk of cardiovascular disease. That's why diets like the Mediterranean diet, which are rich in omega-3s from things like leafy greens, nuts, seeds, and fish, are generally seen as much healthier.
That's a really clear, actionable insight. Okay, now switching gears slightly, but still on fatty acids. The downside of some food processing, trans fats. What happened there?
Right, so for a long time, food manufacturers used a process called partial hydrogenation. They take liquid vegetable oils and add hydrogen to make them more solid, more stable for things like baking or frying, and to improve shelf life. Think margarine or shortening.
Seemed like a good idea at the time.
It did from a food tech perspective, but the process had a really harmful unintended consequence.
Which was?
Well, while it was converting some of the natural cis double bonds into single bonds, it also accidentally converted some of them into trans double bonds.
And trans bonds are the problem.
Yes. So what does this all mean for us? Eating dietary trans fats is strongly linked to increased risk of cardiovascular disease. They tend to raise your bad LDL cholesterol, lower your good HDL cholesterol, and promote inflammation.
That sounds pretty bad all around.
It is. And that's why regulatory bodies pretty much worldwide have now severely limited or banned their use in foods. It's been a significant win for public health, actually.
Good to know. Okay, from storage fats, let's pivot to the structural lipids, the ones that build our cell membranes. You said the membrane is like a barrier.
Exactly. The core of all biological membranes is this double layer of lipids. It acts as a selective barrier, controlling what gets in and out of the cell. It literally defines the cell's edge.
And it forms because these lipids have like two different ends.
Precisely. They're amphipathic. They have water-fearing hydrophobic tails that huddle together away from water and water-loving hydrophilic heads that face the watery environment inside and outside the cell. This dual nature makes them spontaneously assemble into those vital bilayers.
Clever stuff. Many of these are called glycerophospholipids, right? And there's one, cardiolipin, that has a kind of cool evolutionary story.
Yeah, cardiolipin is interesting. It's found mainly in bacterial membranes, but in our cells, it's almost exclusively in the inner membrane of mitochondria.
The powerhouses of the cell.
Right. And its presence there is seen as pretty strong evidence supporting the endosymbiosis hypothesis, the idea that mitochondria actually evolved from ancient bacteria that were sort of swallowed up by other cells billions of years ago and started living inside them.
Wow, so lipid structure hints at ancient history.
Okay.
Then there are ether lipids, slightly different structure.
Yeah, instead of the usual ester bond linking one of the fatty acid chains, they have an ether bond. This makes them resistant to certain enzymes that break down ester bonds.
Any important examples?
Well, one really striking example is platelet activating factor, or PAF. It's an ether lipid, and it's an incredibly potent signaling molecule.
What does it do?
Even at tiny concentrations, it triggers platelet aggregation, which is key for blood clotting. It also plays major roles in inflammation and allergic responses. It's a tiny lipid with a huge physiological punch.
Amazing. And nature uses lipids for environmental adaptations too, like in plants and extreme microbes.
Definitely. Plants, for instance, pack their chloroplast membranes, where photosynthesis happens with lipids called galactolipids. They have sugar head groups instead of phosphate.
Why sugars?
It's thought to be an evolutionary strategy to conserve phosphate, which can be a really scarce nutrient in some soils. Plants are frugal.
Smart. And what about those microbes in extreme places?
Archaea. Some live in boiling hot springs or superacidic pools. Their membrane lipids are unique. They often have these long, branched hydrocarbon chains linked by ether bonds, and sometimes these chains actually span the entire membrane from one side to the other.
Like a single molecule going all the way across.
Exactly. It makes the membrane incredibly stable and resistant to falling apart in those harsh conditions. They're built tough.
Incredible adaptations. Okay, moving on to sphingolipids. You call them complex identifiers? What's that about?
Sphingolipids are another major class of membrane lipids. They're built on a different backbone called sphingosine, not glycerol, but they still generally have a polar head and two nonpolar tails.
And they identify things how?
Well, many of them, especially the ones with complex sugar head groups called glycosphingolipids, are found primarily on the outer surface of the plasma membrane. They act like cell surface markers.
Like name tags.
Kind of. For example, the specific glycosphingolipids on your red blood cells determine your blood group A, B, AB, or O. They're literally the antigens.
Wow. So blood types are lipid structures.
Largely, yes. And more complex ones, called gangliosides, act as recognition sites for signaling molecules or for interactions between cells. So what does this all mean? Their patterns change during development, and even in diseases like cancer. There also targets the cholera toxin, for instance, binds to a specific ganglioside to infect intestinal cells.
And problems with them can cause serious diseases.
Absolutely. Some autoimmune diseases, like Guillain-Barré syndrome, involve the immune system mistakenly attacking gangliosides in nerve cells, leading to muscle weakness and paralysis.
That highlights how crucial they are. Now, if these membrane lipids are so important, what happens when the cell needs to break them down and recycle them, but something goes wrong?
Right. Membrane lipids aren't permanent. They're constantly being turned over, synthesized, inserted, and then broken down. This breakdown usually happens in the lysosome, the cell's recycling center, using specific enzymes called phospholipases. But if there's a genetic defect in one of those enzymes, the lipid it's supposed to break down can't be fully dismantled. The partially broken down lipid accumulates inside the lysosome.
And that causes problems.
Serious problems. These are called lysosomal storage diseases. Tay-Sachs disease is a tragic example, where a specific ganglioside builds up in nerve cells, causing severe neurological damage, usually fatal in early childhood. Niemann-Pick is another group. It's a stark reminder of how vital these disposal pathways are.
Definitely puts it in perspective. Okay, last major structural type. Sterols. Cholesterol is the big one here, right?
Yeah.
What's special about its structure?
Sterols are quite different. They have this characteristic rigid structure made of 4 fused hydrocarbon rings. It's almost flat. Cholesterol is the main sterol in animal tissues. It's amphipathic, mostly hydrophobic, but with a small polar hydroxyl group.
And it does more than just sit in membranes.
Oh, much more. It inserts into membranes and helps modulate their fluidity. But critically, it's the precursor, the starting material for all steroid hormones, like sex hormones, estrogen, testosterone, and adrenal hormones, like cortisol, which regulates stress responses and metabolism, and aldosterone, which controls salt balance. It's also the precursor for bile acids, which are essential for digesting fats in our diet.
So cholesterol gets a bad rap sometimes, but it's actually essential.
Absolutely essential.
Okay.
We just need to keep its transport and levels properly regulated.
Okay, we've covered storage and structure. Now let's shift focus to lipids as active players, signals, cofactors, pigments. Smaller quantities, but huge impact, right?
Exactly. This group might be present in much smaller amounts than, say, storage fats or membrane phospholipids, but they play incredibly dynamic roles in regulating cell processes and communication. They're the managers and messengers.
And some are key intracellular signals, triggering cascades inside the cell.
That's right. A great example involves phosphatidylanoacetyls, or KIs. These are phospholipids found on the inner face of cell membranes. They can get phosphate groups added to them. Now, here's where it gets interesting. When a signal like a hormone binds to a receptor on the cell surface, it can activate an enzyme inside that specifically cuts one of these phosphorylated PIs, let's say PIP2.
It splits the lipid.
Yes. And the two pieces generated IP3 and diacylglycerol act as new messenger molecules inside the cell. They trigger further events like releasing calcium stores or activating other enzymes called protein kinases.
So one signal outside leads to multiple signals inside.
Precisely. It's a signal amplification cascade, a very common and important way cells respond rapidly to external stimuli.
Very cool. Okay, next up, eicosanoids, local messengers.
Yeah, eicosanoids are derived from fatty acids, often arachidonic acid. They act locally near where they're made. We call them paracrine hormones. They don't travel far in the bloodstream.
But they have broad effects.
Oh yeah, dramatic effects. They're involved in inflammation, fever, pain perception, blood clotting, blood pressure regulation, sleep-wake cycles, reproductive processes. I mean, a huge range of physiological responses.
And this connects to everyday things like pain relievers.
Directly. Non-steroidal anti-inflammatory drugs, NSAIDs like aspirin and ibuprofen, work by blocking an enzyme called cyclooxygenase, or COX.
Okay.
COX is crucial for making certain eicosanoids, specifically prostaglandins, involved in pain and inflammation, and thromboxanes, involved in blood clotting. So, by inhibiting COX, these drugs reduce pain, fever, inflammation.
And thin the blood,
Yeah.
which is why low-dose aspirin is used for heart health.
Exactly. It directly targets this lipid signaling pathway.
Fascinating. Okay, beyond local signals, there are the steroid hormones, the long-distance regulators.
Right. As we mentioned, these are derived from cholesterol. They're more polar than cholesterol, allowing them to travel through the bloodstream, often bound to carrier proteins.
And how do they work?
They typically pass through the cell membrane and bind to specific receptor proteins inside the cell, often in the nucleus. This hormone receptor complex then directly binds to DNA and alters the expression of specific genes.
So they change what the cell is doing at a fundamental level.
Yes, they trigger long-term changes in cell function and metabolism, and they're incredibly potent. They work at very low concentrations. Think again of sex hormones or cortisol.
And plants have their own lipid signals too, right? Volatile ones.
Yeah, it's amazing. Plants produce thousands of volatile, often lipid-derived compounds. They use them for communication, attracting pollinators with fragrances like jasmine, repelling herbivores with sharp scents, or even warning nearby plants about insect attacks. It's this whole invisible chemical conversation happening.
Nature's communication network. Okay, another really essential group, the fat-soluble vitamins, A, D, E, and K.
Right. These are all derived from repeating 5 carbon units called isoprenes, so they're often called isoprenoids. They're essential. We have to get them from our diet, and they play incredibly diverse roles.
Let's touch on vitamin D first. It's kind of unique.
It is, because we can actually synthesize vitamin D3 in our skin using UV light from sunlight.
The sunshine vitamin.
Exactly. It's then converted in the liver and kidneys into an active hormone, calcitriol, which is absolutely critical for regulating calcium and phosphate metabolism, essential for healthy bones. Deficiency causes rickets in children. That's why it's often added to milk.
And vitamin A, important for vision, but also other things.
Hugely important. One form, retinol, is the light-absorbing pigment in our eyes. When light hits it, it changes shape, and that's the very first step in triggering a nerve impulse for vision.
Wow.
But another form, retinoic acid, acts as a hormone that regulates gene expression, controlling cell growth, differentiation, and embryonic development. It's so critical. It's even used medically to treat certain leukemias and severe acne.
And deficiency is a major global health problem.
It really is, particularly in developing countries. It's a leading cause of preventable blindness in children and increases mortality. Efforts like golden rice, which is engineered to produce beta-carotene, a precursor to vitamin A, are aimed at tackling this.
Incredible impact. What about E and K quickly?
Vitamin E, mainly tocopherols, acts as a crucial antioxidant. It protects cell membranes, particularly the unsaturated fatty acids in them, from damage by reactive oxygen species.
Okay, a protector.
And vitamin K is absolutely essential for blood clotting. It's needed for a key modification of several proteins involved in forming blood clots, like prothrombin. Interestingly, the anticoagulant drug warfarin works by interfering with vitamin K's action.
So it's both a poison and a medicine.
Depending on the dose and context, yes.
And finally, in this active group, polyketides.
Medical marvels.
Yeah, polyketides are a really diverse group of secondary metabolites, often made by microorganisms. They aren't essential for the organism's basic life, but provide advantages. And many have turned out to be incredibly useful to us as medicines.
Like
things like the antibiotic erythromycin, various antifungals, and even some cholesterol-lowering drugs like lovastatin are polyketides. Nature's medicine cabinet, in a way.
Amazing diversity.
Okay.
All these clear lipids are complex. So how do scientists actually work with them? What are the challenges? Well, the main challenge, as we started with, is their insolubility in water. You can't just dissolve them in a buffer like you would proteins or sugars. You need different techniques.
So, organic solvents.
Exactly. You typically use mixtures of organic solvents like chloroform and methanol, often with a bit of water, to extract the lipids away from everything else in the cell. Proteins, carbohydrates, nucleic acids.
And once you have this mix of lipids, how do you separate them?
Chromatography is key. Techniques like thin layer chromatography, TLC, or high-performance liquid chromatography, HPLC, can separate lipids based on differences in their polarity, how strongly they interact with a stationary material versus the moving solvent.
Okay.
For analyzing fatty acids, gas chromatography, GC, is often used, but you usually have to convert the fatty acids into more volatile forms first, like methyl esters.
And figuring out the exact structure. That sounds tough.
It can be. Traditionally, it involved breaking down complex lipids using specific enzymes like phospholipases and then identifying the pieces. But mass spectrometry has really revolutionized this,
How so?
especially high-resolution mass spectrometry. Techniques often called shotgun lipidomics allow scientists to take a complex mixture of lipids, ionize them, and then measure their mass-to-charge ratio with incredible precision.
So you can identify them directly in the mix.
Often, yes. By comparing the precise masses to databases, you can identify individual lipid species without needing to separate them all first. It's much faster and can detect lipids present in very small amounts. It gives you a detailed snapshot of the cell's lipid profile.
Which leads us to the future. Lipidomics. What's the goal there?
Well, just like genomics aims to map all the genes and proteomics all the proteins, lipidomics aims to identify and quantify all the lipids in a cell or tissue, the entire lipidome.
That sounds like a huge task.
It is. There are databases like lipid maps trying to catalog the thousands upon thousands of known lipid structures. But here's the fascinating part, and maybe the provocative thought to leave with. Animal cells contain well over 1,000 different types of lipids, easily. And for a great many of them, we still have very little idea, or maybe no idea at all, what their specific function is. We know they're there, we can measure them, but what exactly are they doing?
So it's still a lot of mystery.
A huge amount. Lipidomics is opening the door, but there's a vast landscape of discovery still ahead for biochemists and cell biologists trying to understand the full roles these molecules play in health and disease.
Wow. What an incredible journey we've taken through the world of lipids today. From, simple fats storing energy to forming the very fabric of our cells, acting as these critical signal pigments. It's clear they're anything but simple.
Absolutely. This deep dive really gives you, hopefully, a shortcut to appreciating just how central and complex these biomolecules are. We rely on them constantly, often without a second thought.
Indeed. And just think about how much more there is to learn, uncovering the functions of all those unknown lipids in the lipidome. The impact those discoveries could have on understanding our health, treating diseases. It's really quite profound.
It's a field that's absolutely buzzing with potential for groundbreaking insights.
Well, thank you for being part of the Last Minute Lecture family. We hope you gained some valuable insights today.